Hi everyone, this is Evan here to present the Splash course for Spring 2020. It's unfortunate that we weren't able to meet in person, but I hope I can still explain things in a way to you that is clear enough for you to follow along, and then you'll be able to send your questions to me at the end, and I can hopefully give you a little bit of the interactivity that I originally built into this course for you. So this course is called the Apotheosis Hypothesis, and what that essentially describes is the ability for humans to take things, whatever it is that may be troubling us, something in the world that we think is incorrect, problematic, something that we would like to change, and science gives you the ability to make it into something new, to answer the question that nobody else can, and that's really what inspired me to have a career in science, and I hope that I can show you through some of the history as well as through the future work that's being done and the new tools that have been developed, just how powerful it is to have the scientific curiosity on your side when you're trying to solve problems. So a little bit about me. I was born in a small town in New Hampshire called Litchfield, and moved to Boston for my undergraduate study of biochemistry at Northeastern University, where my main activities there in terms of research are studying regeneration and development in the Mexican axolotl, which is a type of salamander that can regrow its arms, its legs, tail, spine even, eyeballs. Pretty incredible stuff. The other work I did was on cancer in metastasis of pancreatic cancer, which is the spread of the tumor throughout the body. It's the main cause of death in cancers. And finally, the innervation of cancer, which is the growth of nerves into those tumors that allows them to continue proliferating, a kind of understudied subject in science. But none of that is what I'm here to talk to you about today. Today, I'm going to focus on more of the type of research that I will be carrying out in the fall in my PhD program for bioengineering at Caltech, where I'm going to be studying synthetic biology and, in essence, the ability to code cells with new functions and really create things that are entirely new, and that's really the focus when I say apotheosis. I mean, these are humans creating things that were previously impossible beyond just repurposing old tools. And I hope I can inspire you to be as passionate about this as I am. And to go a little further into the background, I actually have a video here of me, if I can get it to play. So we're going to skip that video because we're not there in person, but the essence of the video was me as a small child saying that I was going to cure blindness, and that's very relevant to today because actually just a couple weeks ago, lost in the mess of the coronavirus, has been that the first CRISPR trials in humans were conducted in direct injection into the eyes to cure blindness. And this is pretty incredible stuff. CRISPR, for those of you who may not have heard of it, it is very, very hot in the field right now. And CRISPR is kind of a targeted pair of molecular scissors that allows you to home in on a specific gene within a human. So the genes are those functional units that are going to produce a protein of some sort that is going to form a function. So a protein, for example, would be the hemoglobin that allows your blood to carry oxygen. That's encoded in a gene. And what CRISPR does is it allows you to cut like a pair of molecular scissors and change the function of that gene, either delete it, which is being done in this case, or enhance it to be modified in some way to cure whatever deficiency that gene has. And this is just really interesting because CRISPR has never been tested in humans before in a controlled environment like this, where Leber congenital amaurosis 10, which is the type of blindness, is caused by a mutant photoreceptor encoded by this gene, CEP290, and the rest of the cell is healthy. So all they have to do is use these viral vectors that are going to carry in the CRISPR sequence. Here's a little virus for you. And that's going to deliver the sequence into the cell and cut the gene so that they potentially will have their blindness restored. And this is an ongoing study, and so it'll be really exciting to see what comes out of that. But I just wanted to open it up with something that is very topical, not quite as topical nowadays, where the media has been totally taken over by study of the coronavirus. It's warranted attention that it's getting, but this is just a very recent, exciting example to start off with that I will go into a little more in depth later. And again, to tie it back to that idea of apotheosis, I've included some scripture here which references the power of granting sight as something that belongs to the gods. And there's, of course, that iconic scene in Christianity where you have Jesus allowing the blind man to see again. And this is just a very, very visceral, very human need, the ability to see. And it's really ties into the idea of what apotheosis is, that we are now getting to the ability where humans that are born without this fundamental ability can now be brought to equality with the rest of the world. And that's something that I think personally is very incredible, having the ability to give people back something that was unfortunately taken away from them by no cause of their own, other than their genetics. So to get into more of the background of history in which humans have been taking this type of control, again, before we get into the complex tools that exist in the modern times, we'll start with this study here, or this graphic here, representing the evolution of all organisms from about this point here, which would be considered the first life on Earth. And you can see all of this region here, these are just the bacteria in the world, and all the sheer complexity, with each little color fanning out here being one grouping, perhaps a phylum, within the entire within the greater diversity of the world. And then going along this line, you can see the two other domains of the world beyond the bacteria, the archaea, and then here the eukaryotes, and that is just us. That is the sheer complexity of the world that exists around us, and the genes and the way that things exist to survive. This eukaryotic tree contains all of the plants, all of the animals, all of the multicellular organisms, yeast, mushrooms, everything that you can think of. Those all exist within this one tree, which has nowhere near the complexity of the rest of the world. And that's just a bit of context for you as to how minor really the changes that we're able to make are. And when we say that all humans are almost identical, that is genuinely 99.9% of their genes are going to be the same. And the differences we see between each other are just these tiny mutations that occur in singular sequences within our DNA. Maybe a little change here, a little change here, and that can create the incredible diversity that we see in the world. And this graphic on the right is meant to show you that genes can gain new functions through this process of duplication, as just one example, to kind of understand how we can generate the type of diversity that we see there. So when you have a set of genes and they duplicate, so you have multiple of these little blocks that all serve the same function, that means that you can have one of them mutate. So these two in this case, have just mutated, and that may give them a totally new function. And this gives the cell the freedom to invent new capabilities and see if it gives them an evolutionary advantage, where if it's helpful, then it's going to be carried on, and if it's hurtful, then it will end, and that mutation won't be seen anymore. And that's the basic principle of evolution. And this really explains the basic generation of diversity that we see here. This is natural selection, and a process that is totally different from what humans have been doing ever since they entered the scene I don't know exactly the number. I know the recorded history starts about 10,000 years ago, but the human history stretches even further back. As an example of how humans have been biasing this process of natural selection in what is generally termed artificial selection, and kind of molding the world into the way that they would like it to be, again, tying back to that concept of apotheosis, or having some divine guiding power, humans have taken the Brassica oleracea, perhaps I pronounced that incorrectly, but you can look it up there if you would like to, and this plant actually was the origin of among other things, these three different species of plants, or perhaps they're all technically the same species as a result of this, whereby selecting for certain traits, such as the large leaves that have a particular taste to them, or these large buds for an entire head of cabbage, humans were able to generate different types of this plant that have mutations that favor the specific traits that they're interested in. And taking something that is this pretty yellow flower like this, and getting something that is totally unrecognizable but beneficial to us, even if it may not be necessarily the most evolutionarily favorable form for the plant, because we keep it alive, we decide which way it's going to grow. And a similar thing can be seen here with the corn, where corn used to be these tiny little niblets here, about the size of a quarter or twice the size of a quarter, and now we have these giant delicious ears of corn that we can eat, because over years and years and years we picked this corn, and then we planted it because it was bigger than the other corns. And then, we get a bunch of bigger corns, one happens to be bigger, and then we plant this one until we have these large, that's a very bad corn, but picture this as a corn shape, these very large ears of corn that are edible for people, in a way that this original corn plant was not nearly as useful for. And moving on to something that's also familiar to many people is dogs, which naturally we domesticated from wolves. And you can imagine that in terms of surviving in the wild, a wolf is probably going to be the most capable of surviving. However, when humans are taking care of the animal, they can bias it to have particular traits. And on the left here, we have a Labrador Retriever. This is actually my family's dog, Jordy, who was raised to be a guide dog, as you can see for the harness. So he was raised to be a guide dog for the blind people. And this is, so he has particular traits. He has specific obedience trained into him. He's very tolerant, and he's actually trained to have a personality of his own, so that when a blind person tries to tell him to walk into the street, he will be willing to resist them. And then over here, you have my friend's dog, who, by her account, was raised for pretty much nothing except to be cute. The intelligence may be lacking, the danger or the ability to hunt are much lower in these animals. And this is just a very clear example of how humans took something that was natural and branched it off to look the way that they wanted it to look, simply by influencing which ones survived and which ones got to reproduce in a way that interferes with natural selection. And that is the essence of or these are just individual examples of the history of humans trying to control. That's why I title it The Culture of Control. Humans have constantly been trying to control the world around them, and what we're going to get into now is the question of what can we control with the new tools that we have. And that really brings us to the question now of what we would control with the new capabilities of science that exist, the new tools that exist to change things on a molecular level, on the level of their genetics, to try to build things even more faithfully into an image of what we desire than what is being done simply by biasing the survival and relying on those random mutations, inducing specific changes that can solve problems much faster and in a way that nobody ever thought possible. We could cure cancer, we could eliminate genetic disease, we could create and modify memories. And finally, the last example I'll go into is the artificial gametogenesis, which allows for same-sex reproduction, which is something that is important to me as a gay man who has always wanted to be a father of a truly genetic child. So to start in with the curing of genetic disease, of course this is a problem, but I will sell you on it a little bit more right here. 80% of what's classified as rare diseases are going to be caused by some sort of genetic mutation that can be solved in a very simple way, with 1 in 17 individuals being those who are affected by some sort of genetic disease. These can be caused in a variety of ways. We've got pathogenic duplications, deep intronic mutations, point mutations, just 3 examples that I'll cover with 3 very different uses of the CRISPR-Cas tool that I talked a little bit about before, those guided molecular scissors, and how those are allowing us to cure genetic diseases. So one of the earliest, I apologize for the graininess of this photo, but one of the earliest proposed cures is for something that's called Huntington's disease, which is a neurodegenerative disease that has a dominant onset, meaning if you have you have your 2 chromosomes here, and one of them is going to have this H gene. I know that's, sorry, I'll write it over here. One of them is going to be this H gene, and one of those is going to be W for wild type. And if you have just a single one of them, you get this elongated gene with CAG repeats shown here, and that is going to give you these, instead of a nice uniform protein, picture a clumped aggregate like this, due to the increase of those CAG repeats interfering with the way that protein folds, and that causes rapid neurodegeneration and eventually death at around 40. And so this is a very devastating illness that we're looking to solve with CRISPR-Cas9. And the way CRISPR works is it targets a specific sequence, but because the wild type gene still needs these CAG repeats, it just doesn't need as many of them. It's difficult to cut those out. The way that we solve this is that when you have your wild type gene or your Huntington gene, the areas on either side are going to be the same in every single Huntington's patient, because that gene is going to be passed on. That is the cause of Huntington's disease, is this one original mutation, and so it's going to have characteristic spaces around it because of the mechanisms of chromosomal inheritance. You get one from each parent, and if that gene has the Huntington's disease, you're continuing to carry on those sequences around it. That is going to be different due to natural drift of the genomes of humans from what is seen here. And so by targeting the CRISPR system towards these areas on either side, you get cutting out here so that this gene no longer works. And although you're now lacking one of your Huntington's genes, you still have enough of the functional protein here that is not going to be cut due to the extreme specificity of CRISPR-Cas9 cutting. that you'll still see survival in those individuals. And this is a really important possibility that will be coming in the future. And although there are other ways around this, for those who don't want to explore some of the other options of in vitro fertilization, this is a way to enable the elimination of Huntington's disease for parents that may know they're carrying it but don't want their children to have it. Now, going back to Leber congenital amaurosis, we have here, this guy is the photoreceptor in question that has the mutation, and the way this mutation occurs is you have a splice site here. So what splicing is you have introns between the exons, and so this empty space here is not something that you want, so you need to cut it out so you get just this one long mRNA strand that can be turned into a protein. However, what happens in Leber congenital amaurosis is you have a little splicing recognition site here, and so what that causes is a failure to splice where you get something like this. So you only cut part of that intronic sequence out, which is why it's called a deep intronic mutation. And this is going to give you the mutant form of the photoreceptor that doesn't work. What we do in this case is use the CRISPR-Cas system to come in here and cut out that site that's being recognized for splicing, so again we will get the proper form of the gene. And here is just a little model of why this works. So what we're doing, this is the targeting strand here, and how it lines up with the specific sequence using the base pairing principles of DNA and of CRISPR, and it's going to cut at that site. And it doesn't matter that you're going to get a random insertion or mutation here. All that matters is as long as it matches, it will keep cutting, and eventually it will no longer match, and that will solve the problem. Because this isn't in this sequence right here, it isn't in the part that matters, the fact that you're getting a nonsense mutation there isn't actually a problem. And so that's why this disease is kind of the lowest hanging of fruit, and why it was the original target for in vivo CRISPR activity. And on the final front for genetic disease is familial Alzheimer's, and many of you may have heard of Alzheimer's disease or Alzheimer's syndrome, which is when you have the onset of dementia due to the buildup of amyloid beta plaques. I know that was poorly written, and also tau tangles, as a few different signals that signify you have Alzheimer's as opposed to general dementia. And and in the mutation of the gene, or the mutation of this APOE gene into the fourth type, this mutant that has two arginines here, it was found that this is a cause of early onset Alzheimer's, where you get it around 30 years old as opposed to 70 years old. And what we will be able to do to solve this is use what's called dead Cas9 targeted base editing. And this still uses the Cas concept's ability to pair up with the DNA, so you still have that same sequence that matches to specifically target it, but you take the scissors, you break off the tips of those scissors, and instead you attach with a little tether this other protein called the cytidine deaminase. And what that one does is, once it's brought to the site, it flips open the gene sequence, takes the C out, the cytidine, and replaces it with a uracil. And through the DNA repair mechanisms, this changes the base of that DNA, which results in it encoding a cysteine, which you can do then twice at both of these sites, and get a non-mutant form of that APOE gene, which will help to prevent the early onset of Alzheimer's, and that's a really cool possibility as well. Just to give you a sense for how fast moving this field is, the author of this work, David Liu, actually, his lab just published a few days ago a version of this editor that is 600 times more effective, which gives you a sense of not only how fast-moving this is, but also how we're still early in the process, and there is a place for other people to develop better versions and new versions of this if this interests you, specifically because the cytidine deaminase is really the only version of this that we have working right now. There are 4 bases in the DNA. The A pairs with the T, and the C pairs with the G, and all we can do right now is convert C to T right now. And there really is a whole world to explore there in base editing, because it is one of the most precise versions of genome editing. And this is just a really exciting time to be entering into this field, whether you're a chemist looking into the conversion of these, whether you're a bioengineer looking to implement this into a specific control circuit, or whether you're a biologist looking to test some new function or some cure to a disease. Moving on to the disease that everyone has heard of, which is cancer. Affects about 2.5 million people in the United States. That is new cases every year. And there are a variety of solutions to it. Of course, the most common one that people have heard of is chemotherapy, and what that is a high dose of essentially poison to your body that kills all of your cells. And there was bone marrow transplants many people have heard of. What that is you kill all of the cells within the blood of the individual, and then you introduce bone marrow from a healthy individual to reproduce those blood cells, those white blood cells, for instance, that are damaged in leukemia, and allow them to survive once you've killed all of those cells that were infected with cancer. And this was kind of the original example of ex vivo treatment, where you take cells from outside the body, put them into the body to restore some sort of function to it. And the reason I bring that up is because one of the most promising new techniques for curing cancer is what is called CAR T-cell therapy, where T cells are the heat-seeking missiles of your immune system, and the CAR part of that, which stands for cancer antigen receptor, allows this to be a cancer-seeking missile, as opposed to just targeting bacteria or viruses like a normal T-cell would. And how this works is you take the T-cell out of your body, you infect it with these new genetic code that will cause it to express these orange sequences here on its surface, and then instead of just recognizing, like what's seen here, instead of just recognizing the bacteria or the viruses that this T cell would normally target, it's going to specifically find and hunt down cancer cells and destroy them using your immune system without having to kill all of your other cells. And this is a great advantage. Chemotherapy is an incredibly painful and extreme intervention, which in many cases will weaken the individual to the point that they cannot survive for a super long time afterwards anyways. However, these CAR T cells are giving us a new way to hunt down cancers that is a really exciting place to be, and I know a lot of the biotech companies around Boston are currently working on this therapy as a new cure for a disease that pretty much everyone is invested in stopping. And when I say that we are designing custom cells here, these CAR T cells are not just a cell with a new presented surface on it. This would be the surface that recognizes the cancer cell. This little inner loop here is actually what would be used, but we've also modified that receptor to enhance the killing signal that's produced. The release of these cytokines that are going to kill the cell has been enhanced, and the co-stimulatory ligand here refers to the fact that T cells normally need to be activated into a killer form, or there's a cooperation between other T cells. And this, by putting this already on the surface, what it does is it activates the, it activates the other immune cells to initiate their attack on the area, even if they haven't been specifically targeted, and it allows this T cell to be a super killer without needing the support of the cells around it. And that's how we're taking just this one simple concept and then building it into really a biological machine that will do exactly what we want it to do in a way that was possible. And similar to a bone marrow transplant, you transfuse these into the individual, but instead of having to destroy all of their cells first, you're giving them a whole bunch of aid that will help fight the fight that their body is already losing. Now, moving on to something that is getting even more sci-fi, the ability to manipulate memory, and we'll start with the concept of what an engram is. This is kind of a functional unit of memory where when you experience something, you have neurons that are going to fire, and based on if these are all of your neurons, maybe these 3 will fire when you experience something. For instance, when you smelled the smell of your mother's cookies baking at home, something like that. And when you experience these 3 neurons again, that is the engram for that memory, and that is going to give you the feeling of that environment again. That is how you remember things, and that's also why if you smelled those cookies somewhere else, it may bring your mind back to that nostalgic place of your past and your childhood, where the other, so you may have a bunch of other cells, other neurons here, and maybe these 3 remind you of your home. And because these 3 have been activated by the memory of the smell, you're also more likely to remember this by association, and that is the essence of the complexity of brains and memories and neural circuits. And just here is a beautiful picture in pink of the scientists are able to label the cells that are being activated in an engram, and they've labeled this one pink, which means it was participating in their memory, and this one is white because it was not part of the memory that was being trained in the case of this experience. Now, to get into how we're actually going to manipulate and control memories, here we have an experiment that was already done showing that we're capable of deleting memories in mice. And what these blue ones are, they have a death signal. So they have a death signal, and they also have a plus here, which will indicate it is potentiated to become part of an engram. The cells in blue are more likely to become part of that unit. And what that means is when they do fear conditioning, which is a little electrical shock to the feet of these mice, when they hear a tone, that mouse will then associate the sound with the electrical shock, and they will tense up when they hear the sound. And this is basic fear testing, which has been done many times. However, because there is that death signal at the control of some small molecule, when they have generated this engram from the cells they're controlling and then deleted all of those cells using the death signal, they now have a mouse that has forgotten that that tone is associated with a shock. And this has now cured the mouse of what is essentially PTSD to that sound, and there's definitely a lot of potential here to decoupling harmful or anxiety-inducing memories or experiences from people that is beyond just the sci-fi concept of what it means to delete a memory. There are definite advantages that could come here, and although this isn't something that could be done in humans, when you get to the ways of controlling this through optogenetics, which means these neurons are responsive to the light here, with yellow being an inhibitory effect and blue or cyan being a increase in the activity of that engram, you can either repress thoughts, for instance, you in the distant future, you would be able to shine a specific type of radiation on an individual to calm them down if they're panicking, to tamp down a harmful memory. As opposed to, other medication that may be taken right now to stop a panic attack. Or you could overactivate other engrams. For instance, if you get anxiety on planes, as many people do, or if you have a different type of PTSD, a negative association that you'd like to eliminate, you can expose yourself to that environment and then stimulate calming memories to couple those memories together, so that every time you are maybe taking a test that you're worried about, you can eliminate that feeling of anxiety by overlaying it with memories of calm experiences. And this is really an extreme way, definitely the farthest out of what has been covered, of how we could potentially control the human experience itself and make it a more pleasant way when people's brains go haywire and fight against their better interests, taking that control back to the individual so they are in control of their own thoughts, and they're not being overwhelmed by things that have happened to them that they had no control over. And finally, as I mentioned, the original impossible problem that got me interested into science is the concept of in vitro gametogenesis. And what that means is here we have an egg, or sperms, sorry, an egg cell. And when you use an egg donor, for instance, if I were to, with my partner, use an egg donor and a surrogate mother, that would actually mean that only one of us was the parent, and not only that, but that parent would be less than 50% of the genetic information that is in the individual, because the egg is the more important part. It includes the mitochondria and many other key factors that allow for life to grow. And so the contribution of the sperm, although it is still a significant part of the genome sequence, it is less than half, and for legal purposes, this was always very concerning to me. In my understanding of love, I wanted a child that would be half me and half the person that I loved, and I wanted to make that happen no matter what. And it was incredible to me when I learned that we were shockingly close to this being a possibility, where I wouldn't have to worry that this child belonged to someone else more than it belonged to me and my husband. And this isn't a problem that just affects me. There are 48 million infertile couples in the world, and this can also allow them to generate their own eggs or sperm if their own are not functioning properly. And in general, the world is trending towards an increase in infertility, which is somewhat concerning. The levels of impotency are rapidly climbing, and this may become an important problem for everyone sooner rather than later. So to understand how this can be possible, let's just first go back to another really interesting tool that I haven't covered yet, which is the use of induced pluripotent stem cells. And if you aren't aware of what stem cells are, they are essentially they're cells that exist entirely to form other cells. So you may have an amorphous blob cell here. This would be the stem cell, and it has the ability to form either something like a skin cell here, or perhaps a neuron. This is a spindly neuron. I know it's a little bit weird looking, but you've got a skin cell or a neuron. Both of those are part of the ectodermal layer, which means when you're developing, there is a stem cell that's job is to become these two and everything in between within the ectoderm. And the further back you go, in development, all the way back to the original zygote, which can become everything, you have increasing potency. And when you say induced pluripotent stem cells, that means you have a cell that can become anything in the body. And the way that's done is you can take even a skin cell from an adult and infect it with these viruses expressing specific factors, and cause it to revert back in time, essentially making it think that it is back at the start of development. And now, without any sort of, without needing to take any samples from any children or any woman, even in my case, you have a cell that thinks it's undergoing development again. And it has come from me, an adult, or my partner, also an adult. And what that allows us to do is to take those cells and then coax them down their developmental path into thinking that they are developing as a female, and to produce oogonia, which are the structures that form ovaries. And there are naturally concerns when using induced pluripotent stem cells that they do have virus in it. But the reason we go through this pathway is the oogonia, which is spelled like this, undergo a process called erasure, where they delete the parts of the cell that remember what they used to be. And induced pluripotent stem cells do have a memory of what they used to be, so a skin cell would know that it was once a skin cell, even though it has been tricked into thinking it's something else. By getting it to this point, you actually get a perfect, undistinguishable egg cell, whether the host was male or female. And then by providing this egg, as well as the sperm very easily from the other male partner in the relationship, you get an egg cell that, or a fertilized egg, or a zygote, which is a almost perfect genetic product of just the two parents there, which is really incredible to me as someone who cares a lot about children and cares a lot about starting a family. Just having this ability is something that I never thought would be possible, and in fact, this has already been done in mice, and those mice have reproduced for multiple generations, and they're entirely healthy. And we are very, very close to this being possible in humans. So much so to where this originally was what I planned to research in graduate school, but I believe we're so close to it that I can have a totally different career and work even earlier on the fundamental paths of what's possible for solving other people's problems, the same way these scientists have solved this problem for me, kind of paying it on to the next generation and giving them the tools they need to solve their own impossible problems. And then the classic question that I always get, whether it's a joke or not, when I present on my regeneration research at school is, why aren't we doing this in humans yet? And besides the ethical questions of whether this is right, a little bit of what I've been trying to convince you here is that although it may feel like we're playing God in this case, as many would put it, this isn't something that is new. It is more, we have just gained new tools that allow us to control it in a way that was unprecedented. And instead of the dirty mutation-driven control that humans have been exerting, we're doing it at a very precise level that makes things totally new and possible. The only issue with that is these changes are more powerful, and we definitely do have to ask questions. We have to do our best to keep the public informed on what is becoming possible and what type of research is being done, so they don't have to fear what we're doing. And ultimately, the goal is to show that just because it is a difficult decision to make doesn't mean that we should be holding things back from the public, because just as this is kind of an intervention of the natural way of things, it would also be an intervention of its own source to withhold these possibilities when we do have the opportunity to save someone. I don't believe that that's my call to take away the ability to give someone back the life that everyone deserves. And that is what inspires me as a researcher, is wanting to give people the things that they never had, and to level the playing field for a world that too often feels cruel and illogical, trying to make order out of the chaos in a way that can hopefully brighten the world for everybody. And beyond that, why aren't we doing this? Well, there's a lot of work for young scientists such as yourself to be working on. So just to cover the highest level issues that exist with everything that we've talked about so far, gene editing, there's a concern about the lack of precision. You may have heard about the children that were CRISPR'd by a Chinese scientist. The embryos were edited to make them immune to HIV. However, this was really an ethically corrupt decision that was made because the edit he made was poorly designed and very much imprecise. He used a tool that had no business being used at this point in time. And that's why you have people like David Liu that I was talking about with the base editor continuously trying to make it so you have even more perfect control over what's being done. And ultimately with gene editing, because it is something that's passed on to future generations, you need to make sure that you are perfect in what you're doing, because a missed edit is what gives people cancer. Cancer are simple, unknown mutations that become something extreme in their aggregate. And so we have to be careful with the precision there, and that is an ongoing process right now. CAR T therapies, even more extreme. There's an overactivity in releasing these super killers within the cell, and what the next problem that needs to be solved for CAR T cells is how to get them to function within the body without going out of control. And what we've seen a lot of times with CAR T therapies is that they kill other cells as well, beyond the cancer cells. They are so enhanced in their ability to destroy the cancer cells in their attempt to kill all of them that they may end up attacking other cells within your body and giving you autoimmune diseases such as multiple sclerosis and killing you in a whole different way a few years down the line. And the question of whether this is truly better than chemotherapy is really only answered by the potential to improve this therapy. And if we don't keep working on this, then this will have just been another failed attempt to generate a silver bullet for cancer. Memory control, as I alluded to a little bit during that part of the presentation, is inaccessible right now, meaning we don't have a good way to get into the brains of these patients to control this basis. And the reason that we're able to do it in mice is because we have totally edited those mouse lines to be responsive to the treatments that we're giving them. And even in the cases of the light emission studies that were done, that actually requires the opening of the mouse brain and the inclusion of a little LED that's going to release light at our command within their heads, which is certainly not something that's going to be done in humans. And there are a whole lot of advancements that are required before memory control is ever going to be a reality. And finally, as was briefly mentioned, with the issues of iPSCs and development as a whole, there is an incredible amount of unknown complexity that goes on there. And the whole dynamic of knowing whether this recapitulation of development is truly safe enough for the human babies that are going to be generated from it is an ongoing process that we'll have to continue to investigate. And although the erasure process solves some of those problems, we're still very much cheating by growing these iPSCs inside of human ovaries to give them the ability to differentiate properly into the cell type that we're looking for. And for this to ever be possible, there is a lot of unknown complexity in the factors that guide this that has to be solved before we can move forward. And it has been solved in mice, but that is still an ongoing process in humans, although I believe we are very, very close. And Finally, I would just like to open it up to you guys. This was the part of the presentation. I would have gone a little faster if I wasn't making this presentation virtually. So you've been hopefully inspired a little bit by the possibilities, the crazy things that can be done, and the tools that exist out there, of how you would change things, or what is it that you would want to change, what problems you're passionate about. And I'm hoping that I've inspired you to think about that a little bit. If you have anything, any ideas that you want to run by me of potential, or if you want to hear maybe how I would attack a problem that you may not be as knowledgeable of the tools that are out there, I would love to correspond with you and hopefully inspire you to get into this science as well, because I'm always hoping to spread the ideas that are out there right now and kind of demystify the extreme complexity of research that's out there right now. And I believe, yes, here are the citations for all of the information I covered. If you have questions about specific papers or studies, I can link those to you, because this is a bit messy of a slide. There are a lot of sources that were used here. But I would love to hear what you're thinking, and I'm hoping that you guys got something good out of this presentation. And I'm running a little low on time here, but I'm just going to flip to the next slide, and you can pause it there if you'd be interested to see something that I have generated here. This is one of the original projects that I did for a molecular bioengineering course, where I designed a vaccine for HIV, which is the virus that causes AIDS, as many of you may be aware. This is one of the first examples of designing a totally new cell that I worked on, and I know it's very dense, and I can answer questions for you about that later, but I'm running out of time here, and this slide was only included in case we had some extra time, especially in some of the classes where the students, there may not have been as many people to go back and forth about their ideas. I would give you an example to kind of get the ball rolling. So feel free to pause here and try to read this, and I can answer any questions or send you a written explanation if you're interested in it. But other than that, I hope you guys really enjoyed this presentation, and I hope you stay safe right now with the coronavirus. This isn't, just about us. We were sent away. It's not truly a danger to a majority of us, but students such as myself have been sent away from campus because we cannot afford to accelerate the spread of that into the public. And I'm sure you're being swamped in news about the coronavirus right now. The coronavirus actually is a RNA-based virus, similar to what HIV is, which is another reason I wanted to include this. HIV meaning it introduces itself as an RNA and then incorporates itself into your DNA. I believe coronavirus, I'm unsure whether it's a retrovirus or not, but I know that the mechanisms for vaccines are well out there already, and they would be undergoing perhaps a less complex vaccine mechanism than what exists here, but to cover just... But if you're curious, I can also talk to you a little bit more about what a coronavirus vaccine would look like. From my understanding, the main way vaccines work is by priming the body in a similar way to how cancer antigen receptors work. The CAR T cells, you are introducing these little fragments of the virus into the body. For instance, Moderna, which generated the first vaccine, which is currently undergoing testing, uses mRNA delivery to deliver components of the virus which are incapable of replication and infection, but can still be recognized with the body and used to weaponize those heat-seeking CAR T cell, or those heat-seeking T cells, to destroy the virus within the body. And that is the basic mechanism of how a vaccine works. The vaccine for HIV that I've designed is more complex because the HIV directly integrates into the genome and has a extreme level of control that causes it to evade traditional treatments, and that's why this uses some of the tools that are not being used in the vaccines that are being developed right now for the coronavirus. And I know this was a bit of a tangent, but I thought it was important to cover because the coronavirus is the reason that we're not able to have this class in person right now. But I hope you enjoyed, and I'd love to hear your questions.